and welcome to this video podcast for human genetics. In the last video podcast, we focused on the chromosome, its structure, and the parts of the chromosome, and we ended with a brief discussion on karyotypes and how karyotypes can be used and can be compared to determine whether or not a person or perhaps a person's child will have one of many different types of chromosomal abnormalities. In this podcast, I'm going to spend a little bit of time talking about some of the risk factors associated with whether or not a person should be tested, and then I'll talk about some specific tests. So let's go ahead and get started. So on the last whiteboard, we talked about how a cytogeneticist might look at a karyotype to determine the risk of having a child that is going to have a chromosomal abnormality. Now, while a karyotype could be constructed for a person at any stage of life, it does appear that most karyotypes are generated to detect potential problems during embryonic or fetal development. So why would a parent choose to have their fetus karyotyped? In general, a physician would recommend a karyotype of a developing fetus when the risk of having a condition, that is a condition by the fetus, outweighs a risk of a miscarriage. So this is based upon a few things, one of which is family history. Is there a history of some sort of chromosomal abnormality? And the other is maternal age. Typically, this maternal age cutoff is 35 years of age. The procedures to get the cells to be karyotyped, we'll talk about in a moment, but there comes a risk of obtaining the cells to do the karyotype. But we know women over the age of 35 have an increased chance of having a child with one of these chromosomal abnormalities. But why 35? So let me replicate a very common table that we use often to represent why we know age matters for the mom. Over here we're going to have frequency of Down syndrome, which I'll abbreviate DS. And down here we'll have age. And we'll start around age 20, and we'll go 30, 45. We know that the risk of having a child with Down syndrome is about the same from before age 20 all the way to age 30. So it starts to increase at age 35, and it will continue to increase fairly sharply until age 45 and 50. In this range between ages 20 and 30, the incidence of Down syndrome is about 1 in 1,000 live births. At age 35, it's closer to 1 in 500, so the chances have increased quite a bit. And then by age 45, it's more frequent than 1 in 50, which is extremely high. So because of that, physicians, as I said, will usually begin to ask for a karyotype somewhere between ages 35 and beyond. All right, let me erase some of this because I want to keep most of this here. I created a little bit of room here, but I want to ask this question: is why age matters? Because it doesn't matter as much for men. If a man is 35, 40, 45, the incidence of Down syndrome or other chromosomal abnormalities doesn't necessarily go up. And this is largely because a woman has all the eggs she will need at birth. In fact, they're all made during fetal development. So this always blows my mind a little bit, because this means that the egg that eventually was used to make me was developed when my mom was a fetus in my grandmother. So even before my mom was born, in some ways my grandmother was taking care of me because she was taking care of her fetus. I think that's pretty cool. So what's the consequence of that? So what does it matter that all the eggs were made prior to the birth of our mothers? Well, even though they're all made at this stage here, they're paused at prophase 1. And then over this course of 30 to 40 years, those eggs that are paused are exposed to various things that can damage them. So reactive oxygen species is one of those. Reactive oxygen species are known to cause DNA damage, as are viruses that mom may have been exposed to before she had kids, toxins that she may have consumed, and radiation. All of these can contribute to this egg accumulating various kinds of mutations or chromosomal alterations. And we know that that peaks for reasons we don't completely know, but we think it's probably linked to these over here. But that's linked to this increased risk of Down syndrome shown in this graph, but similar graphs could be made with other chromosomal abnormalities. So as we leave this whiteboard, I want to write down a question for you to think about. We may come back to this later. So this is the question I want you to consider. Even though the risk of Down syndrome increases after the age of 35, why is it that over 80% of all Down syndrome children are born to parents who are under the age of 35? It seems a little counterintuitive to this graph I drew here. And there's a few reasons why this statement is true. Like I said, we may come back to this in an activity or a test. Okay, we talked about the risk factors and why we might test. Now the question is, how do we test? And there are two main ways that we can collect cells in order to make a karyotype. And we've mentioned these earlier this semester, but let's talk about them again. So let's begin with an amniocentesis. Guiding a needle into the amniotic sac, cells are collected. So cells are collected from the amniotic sac, or I should say from the amniotic fluid. You don't get a lot of cells, but there's enough cells that kind of fall off the developing fetus. So you get few cells, so that is a disadvantage. So those few cells have to be cultured, and that can take up to 10 days. And the reason you culture them is just so you have more cells to work with. And then from that, the karyotype is made. And then it is analyzed, of course. Using this karyotype, as well as the fluid in general, a physician can help determine whether or not a child will have a chromosomal abnormality, and using the fluid, they can actually measure various biochemical levels. And that can also help determine various diseases that are associated with metabolism. Sometimes, but not often, but sometimes they can detect single gene mutations. But this is usually only if there's a family history. They're not going to test everyone for cystic fibrosis, but if there's a family history of cystic fibrosis, or you know both parents are carriers, then maybe they would check for that single gene mutation. So a couple other things that we should remember about an amniocentesis is that it can't be done until the 14th to the 16th week of development. And then it can be done anytime after that as well. A disadvantage, as I said, it takes a while. So some disadvantages are that it takes a while, but also should include that there is a risk of miscarriage. And so that's why you don't do an amniocentesis for every 22-year-old, because that risk of a miscarriage is greater than the chance of a 22-year-old having a chromosomal abnormality. Doesn't mean it's zero, it just means that the risk is not high enough. We know that using an amniocentesis, we can detect more than 1,000 different known chromosomal or biochemical problems. So it can't detect everything, but it can detect a lot of things. So next, let's look at the chorionic villus sampling, which I'm just going to call CVS. One of the advantages of CVS is that it can be done earlier, at 10 to 12 weeks. So a few weeks earlier. It's also quicker. You get the results back in 2 to 4 days. And the reason for that is with the chorionic villus sampling, a syringe is inserted and cell samples are taken directly from the chorion right here. And so you get a much larger sample of cells. And so you don't need to culture them. So you can just go straight to making the karyotype. However, there are more risks. The risk of a spontaneous abortion or miscarriage is higher. And so that's all taken into consideration when deciding to do a chorionic villus sampling. Though the problem has decreased over the years, there is this slightly increased risk of the child having hand or foot developmental problems. It's rare, but it's still a risk, so it can't be ignored. Now, a question you might be wondering is, how does taking cells from this chorion, how does that tell me about the fetus? So remember, when you were a zygote, this was one cell, and that one cell went through various developmental pathways, and you formed this blastocyst. And remember, the blastocyst had all these cells here that we said was the inner cell mass. And this inner cell mass goes on to become the fetus. The cells around the blastocyst, so remember the blastocyst is like this tennis ball that has this inner cell mass on the inside. But the outer part of this, these are also cells. And these cells go on to form the chorion, the amnion, and all those other structures that we talked about when we talked about human development. So one would predict that all of these cells that make up these extra tissues here outside of the fetus, as well as the cells of the fetus, should all have identical genetic structures. Now, it's rare, but sometimes what happens is that the cells on the outside here that become the chorion, amnion, yolk sac, and all those other things gain a mutation early on and misrepresent these tests. So you could have one genotype, so to say, one chromosomal karyotype from this chorion and a different one on the inner cell mass. So it could, again, it's rare, but it could lead to a false positive result, where you may say the child has Down syndrome, but in reality, the child doesn't have Down syndrome. I also want to talk about ultrasounds. Now, this is an image from a very high-tech 3D ultrasound, and it really gives you a very strong image of what your baby looks like. Because these images are so improved, and quite honestly, a skilled ultrasound tech or a physician could look at those older ultrasounds and they could detect very specific shapes and sizes of structures of the developing baby and could make some pretty good predictions about whether or not the child had some sort of potential chromosomal abnormality. Now, this is rarely taken as absolute proof of a problem, but it will often lead to a reason why you might take the risk of having an amniocentesis or chorionic villus sampling for somebody under the age of 35. Now, the next thing I want to talk about is a less invasive test to determine a chromosomal abnormality. In fact, let's say tests, because there's two things I want to mention here. The first is maternal serum levels. I don't want to spend much time on this, but I do want you to be aware that biochemicals and products made by the developing fetus here can get transmitted across the placenta into the maternal blood supply. So various chemicals. And if you take a blood sample from mom, you can measure the serum levels, measure those various chemicals in that serum, and knowing what should be there versus what is there can lead to a pretty strong prediction of various syndromes that a developing fetus may have. And this has been done for quite some time, and as this advances, at least the predictive level of it, sometimes more invasive techniques such as amniocentesis and chorionic villus sampling are not as necessary. Now, the next one, which is quite honestly really fascinating, is cell-free fetal DNA test. So we know, and we've said this before, that the only thing that can pass through the placenta to go into mom or from the mom into the fetus are various nutrients and other chemicals of small size. So a cell from the fetus isn't going to go across the placenta. However, any cells that die in the fetus, which is common because cells don't live forever, any cell that dies, that DNA that is released from that dead cell, that DNA can move across that placenta. And when that DNA moves across the placenta, it can enter into the maternal blood supply. What this test does is it can detect the fetal DNA. Now the question is, how is it that they can detect the difference between fetal DNA and maternal DNA? Well, if they take DNA out of the blood, the fetal DNA is smaller. And it's not smaller because the fetus is smaller than mom. It's smaller because that DNA that is released from the cells, it doesn't pass through the placenta as this long chromosome. Rather, that chromosome gets busted up just due to the fact that it's not a very stable structure outside of the cell. So what gets released across here are fragments of the fetal DNA. And so any DNA inside of the blood that is mom's will be larger. And so there are various lab techniques to separate short DNA out from longer DNA. And so they've gotten pretty good at, when they're looking at these tests, that they're only looking at fetal DNA. There is some error and there is some maternal DNA in there as well. That would be unreasonable to think you could get rid of all of mom's DNA. But what they do get from here can tell some pretty interesting results. Because what they can do with those small sequences of DNA is they can sequence them. And they can tell very specific mutations, as well as if there is more than predicted of certain regions. So you might expect a certain concentration of DNA from chromosome 21 from a pregnancy that there are no health risks. But if you do the cell-free DNA test and you see an elevated level of chromosome 21 DNA, that would lead you to suspect that the baby has trisomy 21. Now, often after that, then they will go into a more invasive test like an amniocentesis, though sometimes that is the only test that's done. As these tests, as I said before, get better, it will decrease the need for more invasive tests like an amniocentesis or a chorionic villus sampling test. Well, I think that brings us to a nice stopping point for this second video on chromosomes. In this podcast, we talked about various techniques used to collect DNA samples, to collect chromosomes from a developing fetus to determine whether or not that fetus has some sort of chromosomal abnormality. In the next podcast, we're going to build from this and we're going to talk about some specific examples of chromosomal alterations. And we'll talk about how some of those can lead to severe health consequences. In fact, most of them will lead to severe health consequences. If you have any questions over this material, please let me know. If not, I'll see you on the next podcast. Bye for now.